All right. Well, good afternoon. I'm Nick Price. I have the honor and privilege of serving as the Dean and Director here in the College of Agriculture and Environmental Sciences, and so happy to see so many people out here today at the Georgia Center, as well as a number of people that are joining us virtually for today's Brooks Lecture. So this lecture and its associated faculty awards seeks to honor excellence and encourage innovative thought and elevate agriculture and environment here at the University of Georgia, as well as what we do around the world. And we thank you very much for joining us here, as I said, at the Georgia Center. And we have a number of people that are on virtually, so welcome to you all as well. We also want to extend a special appreciation to our D.W. Brooks named professors that we have here in attendance. We have Dr. Peggy Ozias-Akins of the Department of Plant Pathology, Dr. Ignacy Mistel from the Department of Animal and Dairy Sciences, and Dr. Steve Stice of the Department of Animal and Dairy Sciences. Join me in congratulating them. We also have a speaker committee that works on pulling all of this together. I'll call them out. That's Vicki McMacken, Peggy Ozias-Akins, Daniela Lorenko, Alexa Lamb, David Hoisington, and Matus Filipski. So join me in thanking them for their work on making this happen. So today we will enjoy what promises to be a thought-provoking lecture. I'm very sure about that. And we will continue to carry on the legacy of D.W. Brooks, a man who laid the foundation for the work that we do here today. So I'm going to call up this video, assuming everything works like it did before.
The whole idea in the farmer was that I could bring them all up. And I wanted to get them to where they were the greatest producers and the highest income group. I wanted to take them from the bottom to the top if I could.
He would say, never forget who causes things to happen. The workers in the field who harvest the crops, who plant the crops.
I grew up understanding that farmers were the absolute rock bottom supporters of our culture.
One of the stories that really had a great impact on him was what he would tell about his mother back when he was growing up on the farm in Georgia, here, a cotton farm. And his mother would always said, you don't feed people because they're good or feed them because they're bad. You feed people because they're hungry. He saw the great need for something to happen in agriculture. He was a faculty member at the University of Georgia, and admittedly, I think he left that against the better wishes of the Dean of Agriculture at the university and his own father to try to do something to benefit the farmers of Georgia.
The average money that crossed a farmer's hand in a year was $72. He had been to Denmark early in his life and had seen the co-ops there, and he came back and started what was Cotton Producers, which became Goldkist.
The purpose of Goldkist, as Mr. Brooks often articulated it, was to improve the economic well-being of family farmers in the areas it served. Goldkist was one of the first companies that had poultry servicemen whose total function was to go around from farm to farm and help the producers do a better job at what they were doing.
He was absolutely committed to the fact that profits were to be shared, and that's how it happened in a co-op.
Goldkist's legacy in Georgia is that many farm families achieved a much higher standard of living, and their sons and daughters and grandsons and granddaughters continue to enjoy that benefit today through the poultry business. Mr. Brooks influenced agriculture policy very directly. Because Goldkist was the largest agribusiness in the Southeast, he knew presidents and secretaries of agriculture, secretaries of commerce, other cabinet members.
And he served on boards for many presidents, for 7 presidents.
One of the most compelling speeches I heard President Carter make was at Mr. Brooks's 85th birthday celebration at Goldkist. He gave him the credit that he deserved for the leadership that he had provided to Goldkist, to Emory, to the Methodist Church, and to agribusiness in the Southeast.
He loved the university, and that was the only way it was ever referred to in our house. It was the university. When he was in his 90s, he was invited back to give lectures in the School of Agriculture. He loved being able to brag that he had been the youngest professor and now the oldest. One of the things that is so nice about the D.W. Brooks Lecture is that it is an ongoing educational commitment.
I think the importance of the Brooks Lecture today is that it brings the leading thinkers, the leading speakers, the thought leaders to the campus so that young people can see that there are great opportunities in agriculture, agribusiness, agricultural economics, and the environment associated with agricultural activities.
He would be absolutely thrilled about, and he was thrilled about it during his lifetime when he could go. That was one of the highlights of the year.
Today the South is booming, it's doing well, and it's up there even now, and we're doing quite well. So we have great institutions now in the South, and they're training lots of wonderful people, and they're building the South, and that's the way you do it. And so I think the future of the South looks better to me every day.
Okay, I hope that gives you a much better idea about this man, D.W. Brooks. And I noticed that a number of you picked up on the comment that he has the distinction of being both the youngest and the oldest faculty member in our college at the university. Did you also pick up on that too? This is the university. So just to let you know or fill in that gap, he began his teaching career in CAS at 19 years of age, and then ultimately returned to teaching after a long productive career in agriculture that you heard about. And he taught here in our college classrooms till just before his passing, up until the age of 97. So that is quite a distinction that he also holds. We're very grateful for the opportunity that we have here to honor D.W. Brooks' legacy here today, as well as to encourage the pursuit of innovation amongst our faculty members here in our college. I also want to take a brief moment to introduce you to our 2023 winners of our D.W. Brooks Faculty Awards for Excellence, the college's top honor for faculty. Our award winners, I would ask that you please stand when your name is called and remain standing until all winners have been recognized. And our audience, I'd ask you please hold your applause until I've had a chance to call everyone out. So first, we have our 2023 D.W. Brooks Faculty Award for Excellence in Extension, and that goes to Allison Berg, Associate Professor, Registered Dietitian, Nutritionist in UGA Extension Nutrition and Health Specialist in the College of Family and Consumer Sciences. Our 2023 D.W. Brooks Faculty Award for Excellence in Diversity goes to Samia Gray, our Richard B. Russell Chair and Professor in the Department of Poultry Sciences. And I know he's on international travel right now. 2023 D.W. Brooks Faculty Award for Excellence in International Agricultural and Environmental Sciences goes to Woo Kyun Kim, a professor and poultry nutritionist in the Department of Poultry Sciences. We also have 2023 D.W. Brooks Faculty Award for Excellence in Public Service Extension, and that goes to Tammy Cheely, our Ag and Natural Resources Agent and County Extension Coordinator for Glasscock and Warren counties. Our 2023 D.W. Brooks Award for Excellence in Research goes to John Snyder, crop physiologist and professor in the Department of Crop and Soil Sciences, based at our UGA Tifton campus. The 2023 D.W. Brooks Award for Excellence in Teaching goes to Brian Keeper, associate professor in the Department of Poultry Sciences. So we're very grateful for all of your work on behalf of the college. Let's give these outstanding faculty members a round of applause. And also to let you know, we had an awards luncheon earlier today where they were recognized and received their awards at that time. So it's now my honor to introduce you to our guest speaker for this afternoon, Dr. Edward Buckler. Dr. Buckler is a researcher with the U.S. Department of Agriculture's and Agricultural Research Service and an adjunct professor in plant breeding genetics at Cornell University. In his lab, Dr. Buckler uses genomic computation and field approaches to accelerate breeding in maize, sorghum, cassava, and other crops to reduce emissions, ensure food security, improve nutrition, and responsibly respond to climate change. Buckler is a member of the National Academy of Sciences and received the inaugural National Academy of Sciences Prize in Food and Agricultural Sciences in 2017. We are very grateful that Dr. Buckler is here with us today to share his talk entitled Advancing Agriculture, Leveraging Quantitative Genetics and Genomics to Improve Efficiency and Reduce Emission. Please join me in welcoming Dr. Edward Buckler.
Thank you very much. Well, I mentioned this, and I want to reiterate that I had the honor this morning of meeting and talking about with about 50 students this morning. And it was just such a pleasure to talk with them and the great questions they had. And you know, I'm going to talk about a lot of the challenges that we are facing in agriculture. But I think, you know, with the bright people that are coming into agriculture, and if we can continue to recruit the next generation of bright people, these are solvable problems that we can deal with. And so I want to just highlight that, and you know, it's just such a pleasure to meet with everybody today. So the structure of the talk I'll be telling you about is I'll frame it first and what some of the big problems are, and then the second half I'll be telling you a little bit about how those of us working in crops can use some of the most advanced technology to try to address our portion of the problem. But I want to highlight the problem, and to solve these problems are going to take everybody across the entire food system to make this work. So I want to acknowledge that the work that we'll be telling you about has been supported by mostly by the USDA ARS, the National Science Foundation, and now more recently FR and the Grantham Foundation. So the food system is the big problem around it is that essentially we generate about $1.1 trillion of value out of that food system. But as the Rockefeller Foundation has asked, what are the costs of that food system? There's about $1 trillion of costs that come from nutrition and malnutrition and obesity that are in our system. And then we have a little less than about $1 trillion of costs that are coming from environmental and biodiversity losses. And so essentially right now our food system is costing us more than we're getting out of the value of it. And so this is something we need to fundamentally address. And then if you look at it in terms of greenhouse gas emissions, right now agriculture is responsible for about 11% of US greenhouse gas emissions. But by mid-century, we're predicted, because of electrification and all these other sectors, agriculture could be responsible for nearly 40% of US greenhouse gas emissions. And it isn't that agriculture isn't getting more efficient, it absolutely is every year getting more efficient, but it's not decreasing in absolute terms these. And we want to see countries that are kind of like this. This is like Uruguay. They use mostly hydro for power. And so their sector, 60 to 70% of their greenhouse emissions in Uruguay are coming from agriculture. So there's the nitrogen problem, and the phosphorus problem also shows up in our waterways. All through the Midwest, very difficult to do any fishing or anything like that in a lot of those streams, and that's because of that nitrogen phosphorus runoff there. Half of it's coming from corn and soy, where essentially a lot of the pasture that those come off of. And so essentially right now we're at the system where we have a food system that absolutely is the basis of everything that the rest of society is built on, just as Dr. Brooks was highlighting. And you know, it is the most important thing we do is to feed everybody across the planet. But we also need to think about what our environmental impact is and the environmental pollution side of it. And I think there's lots of technologies, but genetics and breeding are key to addressing this. And I'll talk about how I think we can shift the system here. And then that second part of the talk, I'll tell you about how we can solve some of that cropping system side with innovations and concepts we're learning from wild species. So a couple of years ago, a group of us in USDA and OAS were tasked with the question of how do we take on that 38% potential that the U.S. agricultural system could be responsible for 38% of U.S. greenhouse gas emissions. Because I think nobody from a political point of view wants to see agriculture be responsible for 38% of all greenhouse gas emissions or other types of pollution. And so a group of 5 of us then partnering with lots of other people, so everybody from manure management, livestock and range, cropping system, and so on, asked the question, how can we tackle this challenge in a very systemic type of approach? And so the first thing is we went back to the chemistry of life. And just reminding ourselves, this is what agriculture is really based on. It's based on essentially 4 molecules, and then we rearrange them, and we make food out of this. CO2, its big challenge in certain regards is its atmospheric abundance. This is why with burning a small amount of fossil fuels, you can actually have a big effect on it because it's already starting out at such a small proportion of the atmosphere. But this is why plants have to have such massive amounts of leaves is to go collect all that CO2. So the challenge in CO2 from a biology point of view is essentially its concentration, and you really need a massive collector. This is why plants are great at it. They have an insane amount of surface area. When it comes to nitrogen, the big problem with it is to separate those 2 atoms takes a tremendous amount of energy, twice as much energy as is needed for most carbon compounds. But it's an abundant molecule, and this is why industrial processes such as the Haber-Bosch nitrogen fixation reaction were so successful, is because they brought together large amounts of energy with an abundant molecule. Oxygen we don't really have a problem with, and water, its problem is variability. And of course, this is why irrigation, all sorts of other strategies, we are all centered around dealing with variability. So the question is, given those approaches of how to deal with these things, how do we move forward? We've also been thinking about where do we get energy into the system. A decade ago, or 2 decades ago, a lot of us were involved in biofuels, but things have changed. Photovoltaics and other systems have become so cheap, and they take, if you need just electrical energy, they're 25 times better than a plant is getting electrons. And we now can convert that into chemical energy with about 80% efficiency. So this just reminds us that what are plants really good at? They're really good at capturing carbon, and they've actually evolved to have low energy biomass. So where are these problems really coming from? Well, the central problem we think in agriculture is really the loss of nitrogen through the system. We take the nitrogen inputs are about 2/3 from synthetic nitrogen, the Haber-Bosch reaction, where we fixed it chemically, and then the rest from natural fixation. We then lose a little over half of it in crop production. We lose about 1/4 of it in livestock production, and then the rest of it ends up in essentially the consumed crops that, the real target of where we want that nitrogen to go. But before we got there, essentially we've lost 88% of it. Nobody in the system wants to lose 88% of everything you put into it. We want to get it from where we created it to there with the highest level of possible efficiency that we can. And we would argue that, similar to points made by Václav Smil, a technology historian, that synthetic nitrogen really isn't the real problem. It is so much more land and time efficient than biological nitrogen fixation. 2/3 of the globe's population is going to be dependent on it by mid-century. And people frequently highlight how much energy it uses. And they say, oh, it uses 1% of the globe's energy. I would say it only uses 1% of the globe's energy, and you feed 2/3 of the globe's population. That's the most efficient use of energy we're doing. We're doing lots of stupid things with energy that have nowhere near as much value as fixing nitrogen. So I don't think actually our nitrogen fixation itself and doing synthetic nitrogen fixation is the problem. It's how we're taking that very valuable resource that really drives all of life and applying it. Another way to kind of think about that is these are where our big costs are. You can kind of imagine we take urea and other nitrogen sources, we spread them across the continent. We lose about half of that nitrogen between soil, microbes, plants. And then the purpose of that is really to produce crude plant protein. The problem is crude plant protein, that crude plant protein is designed for that seed to germinate the next generation. It's not designed for an animal to consume it and have a balanced diet. This is where in poultry and swine diets and everything, and beef guys know exactly. Those, compared to a balanced amino acid diet, those can be 2 to 3 times more efficient on a per nitrogen basis than crude plant protein is. And so if you combine those two things together, that's where it's only 1/4 to 1/5 as efficient as it. That's where all the losses are. Right through those, because you go through 2 separate steps. Each of them have big inefficiencies. And by the way, human feed efficiency is about the same as these. So it's not any different really than swine. And so we have this greenhouse gas emissions problem. And the nitrogen water pollution is roughly very similar types of numbers here. But this gives you the breakdown of it. And what may surprise you is that in the case of the US, nitrous oxide coming off our fields is the biggest contributor to greenhouse gas emissions. It's not cattle methane, okay? And around the world, cattle methane is the #1 problem, but that's because the diets of many of those cattle are not very efficient, okay? If international cattle were fed as well as US cattle, we wouldn't have anywhere near as much cattle methane coming off of it. So the US system has worked towards efficiency and well-fed animals, and this is the kind of the balance you end up with, is that we're leaking nitrogen off those fields at a tremendous rate, and then again through manure. And so what do we think we can do to change this? Well, fundamentally, we think we should take that nitrogen instead of that we just synthesized and start feeding it to the livestock directly. We, of course, already do this, but generally at relatively low levels. For example, cattle, because they have they can take in urea and other non-protein nitrogen sources, and they can convert that. They essentially have a fermenter, right? And they're able to convert that into amino acids and dry things. For non-ruminants, we need to make amino acids, which we already do, and we complement the grains we provide them. Those amino acids can go on to alternative cultured proteins, or we can also feed them directly to non-ruminants. And so what we're suggesting is that we increase these levels. Of course, we already do this, but 5 to 10-fold. And so what does that do on the plant side? Well, it cuts down the amount of demand for crude protein that we need to produce in the crop side. And so that allows us to essentially recycle the nitrogen on the crop side back into the soil and in the environment. Some plants are very good at recycling nitrogen if you don't take all the nitrogen off the field. And that's fundamentally the key to the problem, is we are pulling too much nitrogen off the field every year. And so that then reduces the amount of nitrogen fertilizer you need to apply. And it also would greatly shift a lot of the manure management side of things. And so just to give you a ballpark idea of the numbers we're talking about, as I mentioned, 5 to 10-fold increases from those base numbers that we're currently giving amino acids to livestock. And that could then perhaps result in a 30% reduction in total crude protein, and perhaps a 50% recycling of the nitrogen that's on the field through that system, and then hopefully at least a 50% reduction in fertilizer input into the system. Another way to kind of look at that is we cut down urea, we cut down the total amount of crude protein, and we make it up on amino acids and urea and other non-protein nitrogen sources. So we're projecting that a model like this could reduce, and this is not something that's going to be changed overnight. These are the types of things that you change 2% per year and you implement over a 20 to 30-year period. But we think we could essentially cut down U.S. greenhouse gas emissions from agriculture by 50% by mid-century. So and I want to highlight that essentially this type of approach requires lots of different technologies to be involved. It starts from everything from modeling and economics, food science, chemistry and fermentation, remote measurement, soil science, and agronomy. But and so I want to highlight that I'm a geneticist and breeder. I'm going to focus in on those types of topics. We don't think that this is a one-dimensional type of problem. It requires all of agriculture to work on this type of problem. So I want to now talk a little bit about the approaches we on the crop side are thinking about. And there it involves things like cold tolerance and nitrogen recycling, which I'll talk more about, but also all sorts of opportunities in new crop rotations and additional perennials in certain environments could play key roles there. And so the first project in this effort that we're kicking off is called Circa, and it's really a keystone effort aimed at corn. And fundamentally, the corn soy rotation is pretty much responsible for about 45% of the problem. Okay, so that's why we're starting with corn because of that centrality in the system. There, it takes half of the fertilizer and everything else. And so what we've done is we've pulled together researchers from actually 30 different labs across the United States to work on this. And we're just getting started right now, or this month. And I want to show you essentially the model we're working off of. Essentially, we're trying to really shift that nitrogen soil cycle. So Mike Castellano, this is his illustration of this model. Our current system is we take a bunch of nitrogen fertilizer, we build up a large pool of nitrate, and that's what the crop taps into. But the problem is that large pool of nitrate is mobile. And so the microbes get it, it leaches, and so we lose a substantial portion of it. What we want to shift to is the diagram on the right, the Circa side, where essentially we're having the pool of nitrate that's needed at the time, but then building up a larger organic nitrogen pool and fast cycling between these so that the crop gets what it needs by keeping down nitrous oxide and leaching off of there. And so the nitrogen fertilizer, then we top off as needed throughout the growing season. Now, there's a very seasonal aspect to this whole thing. Nitrogen in temperate soils like we have here in the US, in the early spring, we actually have a lot of soil nitrate production going on. And the problem is our crops are not growing at that time. And so we end up with annual crops for planting when a lot of that natural nitrate is available. And so it leaches off, or we get nitrous oxide off of it. This is when people talk about cover crops or winter wheat and other types of things having some benefits. A lot of it has to do with this timing aspect of when the crops are on the land. So what we want to do is shift that cycle so that the corn and other crops are able to take better advantage of that natural nitrogen in the cycle and then build up the soil organic nitrogen in the system. And so the strategies we think we can pull this off with is first a couple of strategies that are focused really on nutrient recycling. As we mentioned, the grain protein side, if we are able to get livestock the nitrogen they need from other sources, then we don't need as much protein in the grain. And that mass, although that little nitrogen number there seems small, that is a massive shift in the whole nitrogen balance in the entire system. Because 40% of that nitrogen in the whole system is coming off of corn. We also can then take all that nitrogen that's left over in the leaves and remobilize that nitrogen back down to the roots. That's what every perennial species does at the end of the growing season. When you look at fall colors outside right now, and when it's shifting around, that's what every tree has just done. It's pulled all the nitrogen and phosphorus out of its leaves, and it's sitting in its bark and stem now. And then we can modify the way nitrogen cycles through these processes called nitrification inhibition. Second area is essentially adding more cold tolerance to corn. In the Midwest, we know if we could plant on April 1st instead of May 1st, we've cut down those losses by 20%. And so what that requires is some cold tolerance. We're not talking about getting through the middle of January in Minnesota. We're trying to get it to grow in April in Minnesota. But if we can get down to -5°C temperature tolerance, which we think is probably a tenable trait, not -5 Fahrenheit, -5°C, 20 degrees Fahrenheit, and in places like the Southeast and Texas, that would essentially allow you to have a winter corn crop, something that looked a lot like winter wheat. My colleagues in Texas says he could go from 100 bushels per acre because he gets too hot in the summer to 160. Okay, those are the type of things that if you get a little bit of cold tolerance in the system, you really start shifting things around. And so we are targeting these traits by essentially putting this together, and they work together in order to put together essentially a unified system across here. But I'll also highlight that yes, we'd like to achieve all these traits, but we know we're not going to achieve them all at once and get them out there. But each one of these traits can stand on its own and make incremental progress over the next couple decades. I also want to highlight that I talk about corn. It's not just a corn out there. We truly believe that we really need our land covered year-round with different types of crops. And I think shifting from right now is a lot of focus for cover crop, but what we really need are winter crops. Okay, are really the key thing, so the farmer can make money on both of these things. And that's where we think actually winter legume crops, winter pea, for example, looks to us as a really excellent choice to complement and work with a corn with more flexibility. And so if we could shift a system where we're using winter pea and counter cycle to corn, I think we could produce a much better system. And my colleague Charlie Messina has worked with both Corteva and now the University of Florida, did some back of the envelope type modeling. And essentially, that winter legume circuit corn rotation could increase farmer value at about $250 per acre. So really substantial things out of getting a second crop in there. You're also reducing inputs all through this the system. And so I think that type of double cropping system is what we should really be working towards. So how do we achieve things like this? Well, it turns out that maize has wild relatives, especially perennials that have all these traits. Maize is related to a group of species called the Enteropogonae, and 80% of them are perennials. Tons of them have all these different nitrogen, phosphorus recycling, cold tolerance, and maize even has close wild relatives that have some of these traits. And so what this group is doing is we're trying to learn the mechanisms of how these relatives do these things, and also find the genetics in them to achieve this in corn. And so what I'll talk about for the rest of the talk is really how we can use genomics and AI to go mine these types of approaches out of the germ pattern to achieve these types of goals. So essentially, we've got that system level view, but as you know, if you want to achieve anything, you have to look big, and then you got to focus in on the details and make progress. And so we're going from that cropping system down to how do you figure out how a bunch of perennial species that nobody's ever grown as a crop work. And so this is a question that my colleague Matt Hufford and I focus on tackling. And it's this group of in this group called the Enteropogonae, just to give you a feel. So it's got sugarcane in it, sorghum, miscanthus. These are really efficient crops. And so what we're trying to do is figure out the biology and information technologies, artificial intelligence, not artificial insemination, that's necessary to essentially, I want to figure out if I got a dead plant, but I know what its DNA is, I want to figure out how much genes are working, and then from that, figure out how that organism is adapted to an environment. And I want to highlight that evolution is a really powerful guide to mechanistic models. And we've seen a really powerful example of that just 2 years ago when a group associated with Google called DeepMind developed a machine learning model, an AI model called AlphaFold 2. And it's able to take in a protein sequence, and it can predict the three-dimensional structure of that. And it can do it extremely accurately. This is something that Broward has been working on for 50 years. and they made a jump fold change in just a 2-year period with their latest technologies to pull this off. Is it perfect? No, but it really was so much better than anything we've ever had before. And what they combine, the way they were able to do that is through this combination of using evolutionary history with our knowledge on how proteins fold in their structure. And that combination of putting in the rules of biology or physics and chemistry with evolution seems to work quite well. And so, we're trying to figure out how things work across species. And so, I've got in corn and corn's relatives, 30,000 genes. I got to figure out which of these 30,000 genes do what the heck I want them to do and modify them. And that's fundamentally the question I'm trying to figure out. Which of those 30,000 genes is going to help me make corn adapted to -5C? And so the way we're trying to go about this is we're relying on a lot of quantitative genetics and other types of approaches that we've used to figure out genes and do gene mapping over the last several decades. But we need to start translating into a different way. So 2 decades ago and up till now, what we've been really doing is figuring out what genes are associated with what traits within a species. And the whole growth of GWAS and genetic mapping have been a revolution in that. We can now, if you want, figure out that in a fairly straightforward problem. But the problem with this type of approach is, and we have tools like our practical haplotype graph that allow us to do these types of things and move forward. But the problem with this is in the case of maize, it's a very diverse species. There's 80 million different moving parts and genetic variants that are segregating in a panel of maize varieties. And figuring out which of those 80 million parts matters is really hard. And the next thing is, those genetic variants aren't shared across species. So what you learn about one genetic variant in one species has no bearing on the next species you look at. And so a couple of years ago, we started looking at, how can we start looking at the gene function and putting that into these genetic mapping and genome-wide prediction models. And Andrew Geary did a really nice job of essentially using these imputation approaches where we're able to figure out what the genotype is. And then we combined it with measured transcription levels, and we were able to impute the expression of essentially thousands of lines with those two. And it turned out when she did genome-wide prediction with these, which is the same type of thing, type of model you would use in breeding, she got improved predictions on the order of 5 to 7%. So that suggested if you shortcut the whole thing of using genetic variants, but just started working with trying to infer the function of that gene, how that gene is regulated, you can make progress. Guillaume Ramstein in my group then took on the question of whether he could tackle this with essentially the changes of single amino acids on protein structure, if he could use that to improve these types of models. And it was an interesting way he went about this. He essentially used 2 layers of machine learning models in order to predict evolutionary constraint. And so what he did, and I'll highlight the most important portion of this model was the one called UniRep. It's about protein structure. So he fed the sequences into a model that predicted the changes in protein structure from if you change this one a genetic variant and disruption scores. And then he tuned the model against evolutionary constraint, against a comparison of how that change in protein would be related to how various species are related to one another, combined it with a machine learning model, and was able to then predict how big an impact would be if you changed any given amino acid. And so then he took this problem to go after the basis of heterosis, a basis of hybrid vigor. And so we have useful data from parental hybrids, and he predicted in diverse hybrids, and this predicted nucleotide conservation increased his prediction of yield by 25%. And the neatest thing about this was that inbred line by inbred line, he was able to identify all of the deleterious mutations that were, or some of the deleterious, not all of them. He was able to identify a lot of the key deleterious mutations that were behind that hybrid vigor. And so now, I know that seed companies, some of the major seed companies, are probably using even better models than this, but they're now going in and then essentially identifying the deleterious mutations that are sitting underlying each of their inbred lines, and essentially starting to work to CRISPR them out. So these are the same type of models, if you're not familiar, that people are now using in cancer biology. If you get a cancer diagnosis and you need to figure out what's the cause of that cancer, they're effectively using very similar types of models to this, nucleotide by nucleotide, going through there and figuring out what's broken, okay? And what's broken in the case of crops is the basis of hybrid vigor. Yeah, hybrid vigor is all about fixing all the stuff that's broken, okay? But we now have tools that are essentially being powered by artificial intelligence, machine learning, that let us to go in there. We didn't do any genetic mapping here. We're just looking at the genome and making these connections. So, this, within species, we're allowed to start pulling these together. But what it's given us hope for is that we can start using, because these work within species, but there's no reason we can't run these exact same models across species. So, because they don't rely on us doing genetic mapping, they just rely on us being able to sequence genomes. And so that's what this group, we started a project 5 years ago called PanAN, with some great co-PIs, Chi Sun, Cynthia Romay, Adam Siepel, Toby Kellogg, Jeff Ross-Rivera, and Matt Hufford, where we are aimed to sequence 600 of these species, and together they capture about 1.2 billion years of evolution. And just to give you a feel for that, if we were to sequence 1,000 genomes of corn, that would only give us about 3 million years of total evolutionary time that we'd capture. So this is a very time and an economically efficient way to look at diversity. The neatest things about the Andropogonid, they really are the powerhouse of carbon fixation. They all have the same type of C4 photosynthesis. Those really that evolved about 18 million years ago. In that period of time, they became the dominant species on 17% of the planet. And they have really diverse adaptations to temperature and rainfall. One way to kind of think about it is that when we sequence these genomes, it's like doing saturation evolutionary analysis. Every base pair that started out at the ancestor of this genome has been mutated 50 times by the time we sample it. And we're able to measure, constrain, and then combine these with these machine learning models. So how do you go about sampling these types of things? Well, Toby Kellogg, Taylor Abishon was a technician with her, and she did an amazing job. Started actually, went into the pandemic. During the pandemic, she went around the globe, went to travel, collecting samples for this project, and going into her herbaria and collecting samples there, and getting us thousands of samples that are essentially the basis of this whole project. So this whole project starts with Taylor, and we were incredibly indebted to her. And then my colleague Matt Hufford and Arun essentially worked with Corteva and essentially assembled 40 of these genomes. And these are really complex genomes. The first MAGE genome took $30 million and probably took another $20 million to make it really good. And essentially, but technology's moved on, and we can do this much cheaper. But with this collaboration, these are incredibly complex genomes that we now have, and we can use as our anchors and reference to compare everything against. And so we have things that are hexaploids, tetraploids, all put together, and we can now look at them. And we wanted to fill in the things with lots of those other species. And so we had samples from several hundred herbarium and field specimens, and we did short read sequencing on assembly, and Amy Scholes pulled together these. And we don't have end-to-end sequence for these, but we do reconstruct most of the genes around the genome, and we can compare them to those other ones. And so the big issue we've first started out with is, what are the genes in a species? And this may, if you're not in this field, you may think this is a simple question. Robin Brown has been a pioneer in trying to effectively answer this question of what is a good gene. And I would say the maize genome is not as well annotated as it could be. And this is where we've got 37,000 reference genomes out there, and reference genes in their genome. But there's 1.2 million different possible gene model transcripts that are out there. Which of those are real? Which of them actually get translated into something? Yeah, so this is not an easy question to deal with, but if you're looking for that needle in the haystack that creates perennialism or creates cold tolerance, we need to figure out what's real and what's not real out there. And this is where these AI and machine learning models are really helping us figure out that type of problem. And we can rely on, again, some of the strength of what evolution it focuses in on. Evolution and cells spend 98% of their energy on translation to make proteins. They spend very little energy actually making DNA and RNA. And so selection doesn't really care if you make a bunch of RNA that doesn't get translated. It only cares if it gets translated. Most of the time. There are absolutely lots of exceptions and things like that, but most of the times that's where things are focused in on. And so we created a pipeline called RealGene to pull this together. Jingjing Zhang, she focused on the protein side of the model and essentially started again with another one of those models that was looking at protein structure. This one was trained across all of life. So we took a model that was trained on bacteria and humans and plants and just retrained it for corn, or retrained it for the Enterpogane. And she was able to create a model that said, does this look like a good protein with 95% accuracy? And so the power of these models has gotten really impressive. We created an mRNA model to see whether or not the exon boundaries are correct. We go junction by junction to test those out. And that model had about 95% accuracy, and then one for where does the gene start transcription. That model's not as accurate, it's only 85% accurate, but some of that's biological noise, some of it's probably our data that we're training it on. And so the neat thing was they then took that back to the maize genome and asked the question, does it work? And so we have what we call the maize classic gene set. So these are the very well studied genes. And it said that you got it right 98% of the time. But we actually looked at the 3 errors. It looked like all 3 of those errors are actually mistakes. And the gene got it right, and those 3 were mistakes in the latest annotation of the genome. So we actually think we get roughly at 100% accuracy with this test set for that. But the neat thing was, and we can now differentiate between various classes of genes. And even though there's a lot of these private genes out there, tens of thousands, hundreds of thousands of these private genes, most of them don't matter. Our model does suggest 2% do matter. So those may be really interesting genes. Those 2% that pass this type of filter are the ones that maybe those needles in the haystack we're looking for cold tolerance or nitrogen fixation or nitrogen remobilization. So we've been starting to apply this type of thing to essentially do evolutionary monitoring for perennial genes. As Matt has outlined very beautifully here, the perennial complex is a more complex thing than being an annual. And a perennial has to do about 5 to 7 other things than an annual does in order to make it through its life cycle. Annuals are mostly derivative, in the case of grasses, of perennials. And so there's been losses of essentially suites of genes. And so we use that type of knowledge to ask the question, what genes are being lost in annuals? So what are the genes that are lost or retained in this question? And so it turned out the annual habit, when we started surveying across these group of species, we saw about 80 gene groups the difference in the bar, the dark is that differential gain loss. And in the annuals, there's a substantial increase in loss relative to the perennials. And again, for sequence evolution rate and the premature occurrence of a stop codon, which would stop a protein from working. And essentially what we're seeing across this group of genes is essentially relaxed selection in 3 different areas: development, cell proliferation, cell maintenance. And so now we have, in some of these things, 80 different targets that we can start going after to start asking the question, how do we go modifying these? Shanghai also then looked at the question with these different pairings, looking at just the sister species, what genes were shared between them. And there are relatively few gene losses overall between these things, so which I think helps us focus our search here. And essentially, there were only 23 shared genes between the different contrasts. But out of 30-some thousand genes, the fact that in those low percentages, that was highly enriched. for there. And I think once we look at another couple dozen different lineages, we'll find more overlap and more commonality there. And I hope in a couple more years, we'll kind of understand how you go from perennial to annual in a very systematic fashion, and then we could reverse that as needed. So the next thing we've asked is, we also know that key aspect of how genetics works is you turn on genes at different times, different cells, different time points, and how well can you predict that type of thing? Because that's also really, as everybody in the field knows, you have to get timing right, okay? And that all starts with genes being regulated so they go on and off at the right time. And so in the last couple of years, we've been able to make a lot of progress in machine learning models that allow us to move through those. And I'm not going to go into the details of each of these, but essentially, I'll just highlight that for in the case of we've created models with where transcription factors, the key regulators, they bind. Catherine Mejia and the group developed these types of models, and she created 104 of these models with maze data. She clustered the machine learning models together, and the clusters of machine learning models essentially recapitulated what we knew about this, the genes, related genes in Arabidopsis. This is essentially telling you that the grammar of how transcription factors works is pretty much shared across all flowering plants. And so the lessons we learn, the models we develop in one are really transferable across there. Travis Reichman and the group essentially followed that same line of thinking, working with Bob Schmidt's group here at UGA, looked at whether or not he could model open chromatin regions using data across the angiosperms. And what he found was essentially models trained across the angiosperm were as accurate as a model trained just with the data from one species. So again, the rules are pretty much, for this type of biology, are very much shared. We also found that as we started to sequence these genomes with very high quality and align them very carefully against one another, Armin and Adam Siepel's group, he was looking at individual transcription factor binding sites and how they evolved or gained or lost across the genome. It looks like about 25% maybe lost, maybe a rapid turnover early over. We actually think that may be noise. But for the other 75%, they pretty much get retained for the next 18 million years. So we can pick those out very cleanly. And then finally, we're asking but we know species are being adapted to different temperatures. We have all these temperate grasses, we have tropical ones, and Charlie Hale in the group, he's been correlating where there are various transcription factor binding motifs with these adaptations. And like the CDF2s are correlated with some of those adaptations. And so we now effectively have these tools for pulling them together. So I think we've gotten to this exciting point where machine learning, artificial intelligence are allowing us to predict from the genome is rapidly developing. We can essentially, I think in the next couple of years, go from sequence to predict each of these. Won't be perfect, and it'll get better over the years, but essentially fill in that entire equation. And that essentially will allow us to go mine through species we haven't even grown before. And so that allows you to go from dead plant to how genes kind of work in a predictive fashion. You're going to be wrong sometimes. This isn't going to be 100% work, but enough to generate hypotheses to allow us to start moving forward to understand how things are adapted. And then I just want to end here with how these are all really advanced AI type of models and machine learning. How do we empower other groups to really use these types of tools? And so the last several years, we've been really focused on that merger and getting these things to work together. And I'll just highlight some of the tools we're using right here. But essentially, we are starting to connect things like our practical habitat graph, AI tools into what we call the Breeder Genomics Workbench and the Breeder Genomics Hub. And essentially, this type of tool is allowing people to do repeatable science in notebooks that they're comfortable with, whether in Jupyter, RStudio, and plug in tools that work. And we've started combining these with and deploying these through a USAID funded project to scientists and researchers across sub-Saharan Africa and Latin America. So these tools are open source, available to anybody who'd like to set these up. And then we also, I mentioned at the beginning how important human health was and nutrition. And, these tools that we're talking about can also be applied for addressing those types of questions. And in a project that I'm fortunate to lead, but it's directed by Moira Sheehan with ARS called Breeding Insight, we're essentially taking a lot of these types of genomic selection, other types of tools, and applying them to specialty crops and species around the US. And so over the last 4 to 5 years, they're now expanding that program to support the efforts of breeders across the US and ARS. And in this coming year, they hope to be supporting 28 different species in 59 different programs across the country. So I think these tools, we don't want them to just stay in labs like that, but we really want to expand them across there. So I think we have an opportunity for crossing that off. We're going to have a food system that works and isn't the largest contributor to greenhouse gas by mid-century. I think we can, it's well within our power, but working across these different sectors, we can make this type of thing happen. And working across those, I think we can be. They'll end here. All of the science is done through great collaborations. I mentioned a number of the people who collaborate here. But my group leaders are Sarah Miller, Cynthia Romay, Zach Miller, and Bethany Econopoli. And I'll be happy to take some questions.
Well, thank you, Dr. Buckler. That was a very enlightening presentation, and look forward to having an opportunity for some questions now. For those of you online, please go ahead and type your questions into the chat box. For those of you here in this live audience, please raise your hand. We have our CAS ambassadors that have microphones, and they'll be coming to hand you one. So I think we have a question back here. Is that you, David?
Yes, it is. Thank you very much for a fascinating talk with lots of ideas of how to make agriculture more sustainable and to use less inputs. Now, obviously, that's a key challenge for humanity. And one of the keys to tackling it in an efficient way is dimensioning the problems properly. So I was really refreshed to see that you said that Haber-Bosch isn't the problem, because that's a great misconception within society. Haber-Bosch feeds half the people in the world, so we've really got to hang on to it. I was also really refreshed to see that you said that cows are not the problem, at least US cows, because the size of the US herd has decreased. And there's actually less methane in the atmosphere from US cows than there was in 1970. Because of that. And those stories are stories that the general public doesn't know. But I was concerned about the way that you dimensioned the total contribution of agriculture to greenhouse gases. Obviously, this can be calculated in different ways. But if you look at EPA figures, EPA cites the contributions of agriculture to global gases of the US at 10%. And it also dimensions forestry and land use change to a 12% sink. So one can cite agriculture plus forestry as a 2% sink for the United States. Now that gives a very different impression, and I think it's a impression that is very much true, in that the efficient model of agriculture that America uses is better than the organic model that many people, many urban people want to move to. And that 2% sink, amazingly, is actually without taking into account all of the carbon that agricultural plants fix, all of the grain, all of the stalks, etc., because that's discounted for some reason. So what I'd like you to comment on is that big difference. And I'm worried that when you quote that figure very highly, it misdimensions the problem, and it encourages the urban person, and most people are urban in the States. There's several generations from food production. When asked about what's the problem in global warming, to point their finger at the farmers and not do anything. Perhaps you could comment on those differences.
Yeah, so I think that's very fair to kind of ask the question between net greenhouse gas emissions versus emissions and sink, right? I actually think we should almost always be talking about these both separately, because the accounting side, I mean, I think we need to focus in on emissions and we need to be focusing on sink. I actually don't think we should be talking about net. Okay, we're going to talk about net globally. But I don't think any one sector, because you're right, it's forestry, but then also the amount of climate change we're already baked in for having the next 50 years, those forests are all going to burn. And I think, we need to focus in on stopping the emissions, and then we also need to be focusing on coming up with strategies to sequester as much possible carbon as we can, and we get 2 scores. You get 2 grades. And there's a value for that carbon, and the problem is a lot of that carbon that's out there is not that stabilized. And the soil retention time on it isn't that great, and there's real concerns that, I think we can do a lot better than we are right now. But I'm not convinced that with temperature changes that essentially most forests are going to burn like we're seeing in Canada right now. And we're going to lose these things. And so let's, and we'll have to figure out how to replant, do assisted migration, and all these other types of things to sequester that carbon as soon as we possibly can. But I do think we should keep those 2 numbers separate. And I think it would certainly, I think we can talk about that, but I think I'm not as clear how much carbon we're pulling in. I think we're better at counting our emissions than we are really attributing the land-based sequestration. So. Yes.
Question.
Oh, yeah, I don't need a microphone or anything. I can.
They'll need to hear it online.
Oh, okay. Yeah, great presentation. Thanks. So the take home to some extent is you want to capture a lot of carbon in the soil, and perennials do that. You've known that for a long time, but perennials, you pay a price. You pay a price traditionally in yield. You pay a price in disease pressure. There are certain issues associated with perennials. And so I want to ask you 2 questions. You can decide.
I don't disagree with the first premise. It's not to make perennials, but capture soil carbon. That is not what this model is, Jeff.
Yeah.
This is stealing from perennials, and it's stealing the nitrogen remobilization. This is supported by 5 seed companies because they realize that this is still a very annual-based system that should yield very well.
So you're talking about, when we're talking about perenniality, it's not because you actually want to do it, because there's some aspects of it that are useful rather than the perenniality per se. That's what you're saying.
That's right.
Okay. So let me ask the second question then, which will maybe actually be valid. And that is, if you're going to move earlier planting times, any farmer knows they'll never know when to put the stuff in the soil because it's totally unpredictable. As climate gets more and more and more unpredictable, to what extent do you stick your neck out by pushing that boundary towards the cold part of the year?
Right, and that's where a lot of cold tolerance. I think that's a really good question, and that cold tolerance in many regards is your safety net and your risk. They're already starting to move things forward, but it's risky, and the more you can get, it provides more insurance on it. A lot of the issues also have to do with earlier planting. Temperatures will be getting warmer in these areas on average, but we're also going to get cold spells. Okay, I mean it's going to be both those things, and so that's where that allowing things to move out earlier is going to essentially improve the insurance side of it, and more opportunities for success. But a lot of it also has to do with planting and being able to get out onto the fields, and that's where winter crops and things like that, and then also I think new types of tractors essentially, I've seen these really neat, essentially like a flying wing planter. There's no cab. It's essentially a triangular wedge, and it moves across it. These things are much lighter than anything we move across land right now. And so you can plant on much wetter soil when you're have an automated system that has a planter like that. And my colleagues in Iowa State were showing me this thing. And that's the type of, so I think we're talking about different types of systems with different equipment on it. And so we can get out in, there and plant. And, and sometimes absolutely it's still going to be too wet. You're not going to make August or April 1st. It's going to be April 15th, or it's going to still be too wet. It's going to be, May 15th sometimes. I mean, that's going to happen. So we're not trying to be perfect, but we want to just put that average and that mean in a place that's better. By the way,
Right. So I've got a question about the nitrogen replacement for animal feed and such. So like from a logistics point of view, it makes perfect sense. But we also know that a lot of people in the country, they don't make food decisions based on logistics. There's an emotional component, the strength of the organic industry, all natural and stuff. And so how would you respond to people who have a concern of replacing grains and stuff in livestock diet with what's essentially a protein shake? Because presumably this argument will have to be had at some point. So what is your response to it?
The whole thing is we're already doing all these things already at a different degree. And I would imagine, we're going to run into obstacles on this. And so these are the types of things we're going to change at 1% or 2% per year. And, if there is labeling or other types of things on carbon intensity and things like that, people will kind of see, oh, this has a lower intensity than this other type of product. And then, they may not know why it has it or fully understand the cycle. I mean, I think that the nitrogen cycle is complex. You start looking at what that soil nitrogen cycle is like, I still can't get my head around it as well as the soil scientists can. So I think, there will be some people who are resistant to it, but I think the economics long term would favor this efficiency. And so I think some consumers will say, well, it's cheaper, it has lower carbon intensity, and I'm going to go that way. And other people are going to make another decision there on that. And I would also kind of highlight, I didn't say what percent I think people are going to go for alternative proteins versus animal proteins. That's the other big battle in the market. And I have no idea where the consumer is going to average out in that type of system. And I would rather us try to fix the entire system and not try to make a bet one way or another on where the consumer is going to go in the system. So I'm not quite sure how to deal with it, but that's my thoughts on it.
I have a question about the selection of cultivars because, well, to increase efficiency, we are breeding plants and also we are breeding animals. And I know a little bit about plants and I know lots about animals. So in the animal field, there was an effort 25 years ago to find gene responsible for this. And the effort failed completely, I would say. And now breeding companies are not using genes, just using similarity across animals. Mm-hmm. And then, well, simply because all good genes, or nearly all of them, have already been fixed. And those who are left have kind of good effect on one trait, detrimental on something else. So the question is, with 30,000 genes or something like that, how easy will be to have kind of a useful model for efficient selection?
Yeah. So I think in some of the traits we're talking about here, for example, I think that the cold tolerance trait that we're going after, it may be really highly complex and quantitative. Okay, I hope not, but it may be, and that type of selection model may be exactly the way we would need to deliver it. And it may just be too hard to move it into a species like corn. And that's why we go with things like, wood pea and a different type of rotation system rather to get us the same ecosystem product out of it. But I think we can parameterize some of these models and be able to move through introgression relatively rapidly. And I would say for these deleterious mutations I was kind of talking about for hybrid vigor, from what I've been hearing in the industry, their efficiency is high enough that, and the value is worth, even though it's a quantitative trait, to purge them. And you can edit fast enough now and do multiplex editing enough that it matters. And it's cost effective now. So, so some of these things aren't going to be one gene. It may still be 20 or 30 genes, but we have the technology now to go after 20, 30 genes or 100 genes at once, especially if they're natural alleles. You're not introducing a new gene, you're fixing a gene that may have been lost or something like that.
Sorry.
Fascinating talk. And I work with wild, so it was mind-blowing seeing the AI and wild species. So I think my question is, is a leeway from what's asked before. So AI is just, any model is just as good as your data points are. Whatever feeds your model is going to make it efficient or not.
Right.
And you get information from very diverse wild species from different genus, genera. And a wild species that we know, it doesn't really prioritize yield. That's something that we prioritize, but not the wild species. So I was surprised when what you showed as a response to your model is productivity. And we know that genome, sequencing a genome now is cheap, but phenotyping is actually the key to getting any data, meaningful data. So I'd like you to elaborate a little bit how useful your model is going to be for maize or anything else. How did you get information from sugarcane and whatever else for maize?
So I think one of the key things, at least for grain yield, it is somewhat fairly closely related to fitness, okay? And this isn't true for a lot of other traits. I mean, it isn't a one-to-one match, but there is, which is a lot of other traits, there's a fairly strong correlation between fitness and grain yield in corn. And so that kind of lets us somewhat cheat evolutionary model, because what evolution was selecting for is somewhat similar to what we're trying to select for there. It's not, so I think that's part of it. And then I would highlight in almost all these things, yes, we train, the massive number of data points are coming from all these different species, but then there's always a retraining and optimization of the model that's specific for that species. Okay, so yes, we trained, identified, and prioritized the weightings based on all bacterial protein structures, but then the model still gets re-optimized on yield data that's been, several thousand lines and hybrids that have been trialed, and that model then gets optimized, and then we use that for those predictions. So it's not, I don't feel like we're not really, there's a lot of information that gets into prioritization, but then the essentially the weighting for the physiology needs to be more species specific. Okay, so I think we can get to, and this is where I think biology is really shared, a lot of biology is really shared up to the level of translation. Okay, and across the Andropogane, it may be all the way shared across the synthesis and a lot of development or something like that. But that doesn't mean it still gets you necessarily to yield. And so once you get down to that final aspect of yield, you've maybe reduced the dimensionality of the problem with these other surrogates that you can use across species, but then you still need to do some training, others. We cannot look at a genome sequence and tell you what the yield and adaptation is going to be with no within species data. But I think we can tell you, we'll be able to say that this gene is turned on at this time in development, and roughly at this level relative to other alleles out there or other species versions of it. And from that, you can relate that to adaptation. So I think that's a tenable thing, but not exactly how it's going to be adapted. So I think we can get these relative rankings within clades like this, but not some absolute level of adaptation.
Okay, I'll ask one last question.
Oh, sure.
So based on this work, when do you see us growing corn in northern Minnesota in early March?
In early March. I was trying to get to April. We'd like to make it through Seth Marley. He's in Texas A&M. And he'd like to have overwintering before he ends his career. And he's about 10 years younger than me. So I think he'd like to see what we can do to get corn to overwinter in East Texas. So that I think is a doable target type of thing, if this is tenable.
That'd be a huge accomplishment for sure. So, yeah. Join me in thanking Dr. Buckler for being here for his presentation.
Thank you.
So as a gift of appreciation, we have a box of Georgia grown products here to give you. We will send those to you so you don't have to take them. But it's a whole host of things that we're sure that you will enjoy here from the great state of Georgia.
Thank you.
So thank you very much. Appreciate it. So in closing, this is always a very special day for us here in the College of Agricultural and Environmental Sciences. And this is a day when we have an opportunity to step back and reflect on all the excellent work that we do, that you all do, and to gain inspiration from our guest speaker for ongoing and future initiatives. And congratulations again to all our D.W. Brooks Award winners. Thanks to each and every one of you for joining us today. And thank you for all that you do on behalf of the college as well as the university. Remember that. All right, let's enjoy the rest of your day.